Glycolysis is the process of breaking down glucose into pyruvic acid. It is the first stage in cellular respiration. Glycolysis occurs within the cytoplasm of the cell. It begins with the first step when hexokinase transfers a phosphate group from ATP to glucose, producing ADP and glucose 6-phosphate. In the second step, phosphoglucoisomerase is added to the molecule glucose 6-phosphate and converts it into its isomer, fructose 6-phosphate. Step 3. Fructose 6-phosphate gains another phosphate by the use of an ATP molecule and the enzyme phosphofructokinase. This creates ADP and fructose 1,6-biphosphate. In step 4, fructose 1,6-biphosphate is split into two different sugars by the enzyme aldolase. The two sugars are dihydroxyacetone phosphate and glyceraldehyde phosphate. Step 5 begins when the enzyme triose phosphate isomerase quickly converts dihydroxyacetone phosphate into its isomer, glyceraldehyde phosphate, resulting in 2-glyceraldehyde phosphate. In the sixth step, the 2-glyceraldehyde phosphate are combined with the enzyme triose phosphate dehydrogenase. This enzyme transfers hydrogen from glyceraldehyde phosphate into NAD+ to form NADH. Next, the enzyme will take a phosphate from the cytosol to each glyceraldehyde phosphate to form 1,3-bisphosphoglycerate. By step 7, there are 2,1,3-biphosphoglycerate. The enzyme phosphoglycerokinase transfers one phosphate from each biphosphoglycerate to a molecule of ADP to form ATP. This produces 2 3-phosphoglycerate molecules and 2 molecules of ATP. In step 8, the enzyme phosphoglyceromutase takes a phosphate from each 3-phosphoglycerate from the third carbon and transfers it to the second carbon to form 2-phosphoglycerate. During step 9, a molecule of water is removed from each phosphoglycerate by the enzyme enolase to form phosphophenolpyruvic acid. In the final step of glycolysis, the enzyme pyruvate kinase transfers a phosphate from phosphophenolpyruvic acid to the ADP to form pyruvic acid and ATP. The final products of glycolysis are 2-pyruvate and 2-ATP. Formation of acetyl-CoA begins when the pyruvate made from glycolysis travels from the cytosol into the mitochondria. Formation of acetyl-CoA is also called oxidative decarboxylation. The enzyme used in this process is pyruvate dehydrogenase. First, a carboxyl group is removed from pyruvate as carbon dioxide. NAD removes a hydrogen from pyruvate for the formation of NADH. Finally, the oxidized carbon fragment attaches to the coenzyme A, forming acetyl-CoA. The remaining products of this process are acetyl-CoA, NADH, hydrogen, and carbon dioxide.